the world's deadliest infectious disease. One name continues to dominate the list, a disease that claims over millions and millions of lives, leaving behind a trail of mortality, morbidity, and immense human suffering. Yes, we are talking about tuberculosis. Despite being preventable and treatable, TB remains a global health crisis, the world's deadliest infectious disease. Tuberculosis now has taken over millions of lives. By 2023 only, there's been 8.2 million of new cases, and the WHO has stated that this data is placing TB again on the leading infectious disease killer, suppressing even COVID-19. More alerting reports found about 845,000 cases reported in Indonesia, with over 13,000 deaths that we failed to prevent. But what is the problem? What are the persisting gaps, and why is our so-called advanced medical world still stuck in these dangerous numbers? Well, the existing bacille-calmette-guerin vaccine, or as we know, the BCG vaccine, a century-old tool in TB prevention, is no longer enough. Its inconsistent efficacy, particularly in equatorial regions like Indonesia, leaves millions vulnerable. Meanwhile, the growing threat of multi-drug resistant TB adds a new layer of complexity to this crisis. The growing threat of multi-drug resistant tuberculosis underscores a critical gap in our fights against TB. So, when we have increasing resistance to the standard treatment, MDR-TB possesses a significant challenge, complicating care and amplifying the urgency for innovative prevention and also control measures. So, for this critical matter, we gathered an amazing multidisciplinary team from Faculty of Medicine and Faculty of Biology, also collaborating from different universities, Universitas Indonesia, Gajah Mada, Jenderal Sudirman, and Pajajaran, including Rafli, Zainab, Ode, Renada, and Zaki. We studied our possible insight, tackling with a start into computational analysis. Here, we bring the title, Novel Multimer Chimeric Vaccine Candidate for Tuberculosis, a comprehensive in-silico study with AG85 protein complex on Indonesian population. By leveraging advanced computational methods and tailoring our design to the unique genetic profile of Indonesia's population, we aim to fill the gaps left by BCG vaccine and offer a strong, innovative approach to TB prevention. This is not just a scientific pursuit, it's an urgent necessity. Now, next on, let's take a look on the methodological approaches we used for our study.
Here's the design of the method used. First, we place homologous sequences from various microbacterium tuberculosis protein sequences in the same column through multiple sequence alignment. Then, the results were aligned using the analysis of conserved sequences. Afterward, the protein sequences obtained were subjected to a bit of generation, including prediction of MHC class 1 and class 2, also B-cell epitopes. Then, the epitope candidates were analyzed for immunogenicity, including antigenicity, allergenicity, and toxicity assays. Only epitopes that have antigenic, non-allergenic, and non-toxic properties are further screened within the population using population coverage. The selected epitopes were designed into a vaccine construct with the addition of large ribosomal subunit protein called BL12 as an adjuvant. Furthermore, the vaccine constructs were analyzed for immunogenicity and physicochemical characteristics, including molecular weight, theoretical isoelectric point, instability index, aliphatic index, and the grand average of hydrophobicity. Then further analysis was done to see the homology matching with human peptides and gut microbiome. Non-homogeneous considered when F values less than 0.005 or the identical percentage was less than 50%. The vaccine models also analyzed for immune response using ChimSim server, and the simulation parameters were kept to the default, except for the time steps in 1.84 and 170 with 1,050 of total simulation phases. Hence, there were 2 injections at the interval of 4 weeks. The interaction between the vaccine and TLR4 protein was tested using molecular docking with HDoc software. And lastly, for prospecting the large-scale production, we also constructed plasmids for cloning of the model vaccine.
Here, we present the results of multiple sequences alignment and conservancy analysis, detailed in the supplementary folder and summarized in Table 1. This analysis focused on the antigens AG85A, AG85B, and AG85C, identifying conserved peptide sequences across all 3. As shown in Table 1, these conserved sequences are critical for designing effective epitopes for the vaccine. They enhance the likelihood of eliciting a strong and consistent immune response due to their stability across various strains. The result of the epitope selection process are summarized in Table 2. As shown, all selected epitopes meet the essential criteria. They are non-allergenic and non-toxic. They exhibit strong antigenicity, ensuring a robust immune response. Each epitope binds to at least 2 or 3 alleles from the Indonesian human leukocyte antigen alleles, enhancing population coverage to create herd immunity. The vaccine construct combines 3 B-cell epitopes, 3 HTL epitopes, and 3 CTL epitopes. To connect these components, we use specific linkers, GPGPG for HTL epitopes, AAY for CTL epitopes, GGGS for B-cell epitopes, KK and EAAAK for incorporating the adjuvant and ensuring structural integrity. The large ribosomal subunit protein BL12 from Mycobacterium was included as an adjuvant at the N-terminal. This design improves the vaccine antigenicity and enhances its ability to stimulate a robust immune response.
Using data on the most common HLA alleles in Indonesia, we selected 3 epitopes from class 1 and 3 epitopes from class 2. The analysis show that this combination provides 97.61% population coverage, which is extremely high and close to the ideal range needed to create herd immunity. This means the vaccine could potentially protect nearly the entire Indonesian population. In addition to immunological compatibility, the physicochemical properties of the vaccine construct were analyzed. The molecular weight was found to be 29.8 kilodalton, an optimal size for uptake and presentation by antigen-presenting cells. Moreover, its isoelectric point, or PI, of 4.78 is well suited to maintain solubility and stability in physiological conditions, particularly for injectable formulation. Another key strength lies in the vaccine's structural stability. With a high aliphatic index of 79.44, the construct demonstrates excellent thermal stability, making it suitable for varied temperature conditions, a critical advantage for distribution and storage, especially in tropical archipelagic countries like Indonesia. Additionally, a negative gravity score signifies that the vaccine construct is hydrophilic, further enhancing its solubility in aqueous environment, which is important for formulation into injectable solution. The estimated half-life of the vaccine in different cell systems confirm its robustness and adaptability across mammalian, yeast, and bacterial systems. This makes the production process more versatile and efficient. We also analyzed the homology with human peptide and gut microbiome to ensure vaccine safety by avoiding components that could mimic human protein or microbiome element, which might cause adverse immune reaction or reduce effectiveness. In this study, CTL and HTL were analyzed for similarity with human peptides and the human gut microbiome. A non-homologous assay identified 9 mer sequence, which are the core part of the peptides, interacting with HLA and T-cell receptors. The result show no 100% match between the selected 9 mers and human peptides, and no significant similarity to the gut microbiome. These findings confirm that the vaccine construct is safe and will not disrupt the immune balance of the host. The immune response from vaccine candidate were built upon the initial injection, with the second and tertiary injection having a greater increase in immunoglobulin and immunocomplex, like in figure 2a, B cell population in figure 2b, and T helper population in figure 2c. Immune simulation also indicate the involvement of a possible innate and cytokine-based regulation, with macrophage being active and increasing IF and gamma level for a certain time period. IF and gamma itself play a key role in tuberculosis defense by activating macrophage to produce reactive nitrogen species like nitric oxide. Protein-protein docking toward the medium of the immune also demonstrates strong vaccine binding affinity to TLL4, forming multiple hydrogen bonds and nonpolar interaction. The 3D visualization, as you can see, confirm robust interaction at their binding sites. Binding affinity were notable around minus 275.60 kilocalorie, and that's enough for vaccine. Many studies has already shown TLL4 expression correlated with protective immune against Mycobacterium tuberculosis. The vaccine construct was then modeled into the PET21 plasmid using T7 RNA polymerase expression system, resulting in around 6,000 base pair construct. This system facilitated efficient protein and DNA expression.
As we reach the conclusion of our study, we are thrilled to present a breakthrough in TB vaccine development, in our case, tailored specifically for the Indonesian population. Our Multimer vaccine construct, designed with dominant HLA alleles, achieves a remarkable population coverage of 97.61%, as it was mentioned before. This not only underscores its precision, but also its safety and reduced risk of adverse immune reactions. This vaccine is built with broad-scale immunization potential, making it a strong candidate to combat TB within Indonesia and even beyond. With its high immunogenicity and safety profile, it serves as a beacon of hope in global efforts to eradicate tuberculosis. If successfully developed and implemented, this vaccine has the potential to drastically reduce TB cases, becoming a cornerstone in prevention and also control strategies. Beyond Indonesia, it sets a precedent for other nations battling similar challenges, illustrating how targeted vaccines can reshape our public health. However, this is just the beginning. To unlock its true potential, we must move forward with clinical trials and wet lab validations to ensure its scalability and efficacy for real-world application. Now, how can we turn this vision into reality? So the next phase requires recombinant DNA technology to express and purify the multimeric protein construct, and this will pave the way for crucial in vivo studies. We're employing animal models like, for example, mice or guinea pigs to assess the immunogenicity, the safety, and also the efficacy. Another important point is refining vaccine formulation through adjuvant selection and stability testing is really essential to prepare the candidate for clinical trials. So these steps are very critical to bridge the gap between theoretical promises that we have and the tangible impact that we want. And by ensuring that every aspect of this vaccine is tested and optimized, we can translate this innovation into a game changer for tuberculosis eradication. In closing, the fight against TB is not just a medical challenge, it's a global imperative. This vaccine candidate symbolizes the power of science and collaboration to address the unmet needs of millions and millions. We have the tools, we have the knowledge, and the opportunity to rewrite the story of TB in Indonesia and around the world. So together, let us take the bold steps necessary to turn this potential into a life-saving reality. Thank you.